Hello everyone, myself Shagor Cham. Today I am presenting BioLLMNet, a unified sequence-only framework for multimodal RNA interaction prediction, authored by Abrar, Rahman, Abir, MD Toki, Tahmid, and Shamsuzzuha Bayzid. The central idea of this work is to predict how RNA interacts with proteins, small molecules, and the other RNA using learned sequence representations rather than relying on difficult to obtain structural information. This makes the method both practical and broadly applicable to several important biological problems. The interaction bottleneck. This slide highlights the main biological and computational challenge. RNA interactions are extremely important in post transactional regulation and disease, but traditional prediction methods are limited because they depend heavily on structural data, physicochemical properties, and manually engineered features. In real settings, those inputs are often incomplete or unavailable. So the problem is not just about accuracy, it is also about building a method that remains useful when experimental information is missing. Moving beyond handcrafted structural dependency. Here the paper makes its conceptual shift very clear. Instead of requiring sequence plus 3D structure, BioLLM networks with sequence only inputs and uses pre-trained biological language models to extract richer representations automatically. The value of this approach is that it reduces dependence on expert crafted biological features and moves toward a more general framework that can be applied across different interaction partners. The BioLLMNet paradigm. This slide explains the broader vision behind the method. The author treat RNA, protein, and small molecule sequels almost like different biological languages, each encoded by a specialized foundation model and then unified inside one prediction framework. The important point is that BioLLMNet is not just one model for one task, it is designed to support multiple interaction types under a single architecture, which is a major strength of the paper. A unified three-stage architectural blueprint. The architecture follows a simple but powerful three-stage pipeline encoding transformation gated fusion first. First, each biological sequence is converted into a pre-trained embedding. Second, the embeddings are mapped into a compatible feature space. Third, the model combines them in a way that depends on the input context rather than using a fixed merging rule. This staged design is important because it keeps the framework unified while still respecting the differences between modalities. Step 1, encoding biological syntax by foundation models. This slide shows the first stage of in more details. RNA is encoded using Renalmo, protein sequence are encoded using ESM2, and small molecules are encoded using MOLBERT. Each model captures modality-specific biological meanings. Renalmo provides a 1280 dimensional RNA representation. USM2 produces a 1024 dimensional protein embedding, and MOLBERT gives 768 dimensional molecular embedding. So instead of handcrafted design features, the model begins with rich pre-trained representations. They already content contextual biological information. Step 2, cross-modality feature transformation. Here the problem is alignment. These embeddings come from different models and therefore live in different vector spaces, so they cannot be fused directly. BioLLMNet solves this using a learnable fusion trust layer that projects the RNA encode embedding into the dimension of the interaction partner. That's that, step is subtle but essential because it allows the model to compare between heterogeneous biological signals in a mathematically consistent way. Step 3, adaptive information integration via gated fusion. This is the more distinctive part of this paper. Instead of simply concatenating the two embeddings or averaging them equally, BioLLMNet learns a gated that assigns feature-wise weights to each modality. So for one interaction, the RNA signal may matter more. For another, the partner signal may dominate. That makes the fusion dynamic and contextual, which is exactly why the model can adapt across different biological tasks. Comprehensive empirical validation across three modalities. This slide summarizes the evaluation strategy. The author tests the framework on three different distinct settings: RNA prediction, protein interaction prediction, RNA small molecule interaction prediction, and RNA-RNA interaction prediction. This is important because it shows the model is not narrowly optimized for one benchmark. The paper is really asking whether one architecture can remain strong across multiple biological interaction families, and that is what makes the evolution convincing. Setting a new benchmark for RNA prediction interactions. On the RNA protein task, BioLLMNet delivers one of its strongest results. On the RP1460 benchmark, it outperforms the best baseline RNA encoder across the major metrics including MCC, accuracy, F1 score, precision, recall, and AUC ROC. The key message here is not just the model is better, but the improvement is broad and consistent, which suggests the learned representations are capturing meaningful interaction patterns. Systematically lower error rates across RNA subtypes. This let's focuses on RNA small molecule prediction. BioLLMNet achieve strong Pearson correlation and lower mean absolute error across multiple RNA subtypes groups, which means it is not only predicting trends correctly but also reducing numerical prediction error. The model is especially effective on challenging categories such as ribosomal RNAs, and reported binary classification results also showing very high MCC scores, including a particularly strong result for viral RNAs. Retaining superiority within a shared language. Here the authors ask an integrating question. Does the same framework still help when both sequences come from the RNA domain? The answer is yes. In the miRNA lncRNA setting, BioLLMNet remains competitive and several cross-specific settings it outperforms previous methods, including a notable improvement on the MTR-ATU split. This shows that the architecture is not limited to cross-domain interaction prediction. It also generalizes within the same biological language. Case study mapping the 2QEX0 RNA interaction hub. This case study makes the performance easier to interpret. Biologically, the RNA sequence has 37 interaction partners, so it is a strong example for testing real-world usefulness. BioLLMNet correctly identifies all 29 true interactions within the 2QEX family and makes only two false positive predictions, which gives it very strong recall while still keeping a balanced overall profile. The architecture is the breakthrough. This led one of the most important takeaways of the presentation. The paper shows that the performance gain does not come only from the using language model. It also comes from the way the embeddings are integrated. Concatenation performs the worst. Element-wise averaging is better, and gated fusion performs best across the all major tasks. To, to conclude, BioLLMNet presents a unified sequence-only framework for predicting RNA interactions with proteins, small molecules, and other RNAs. Its main contribution is that it replaces handcrafted modality-specific pipelines with pre-trained biological embeddings and a learnable fusion strategy, which improves both generalization and practical usability. The paper also points to future directions such as adding cross-modal attention, joint multimodal pre-training, and even 3D molecular structure information, which could make the framework even stronger in next version. Overall, this work is a strong example how biological language models can reshape interaction prediction. Thank you.